Hello everyone, welcome to Biological Sciences. This video is a quiz video which is related to the upcoming exam CUET 2022. Here I have bring you some questions related to biology and biotechnology. So here is our first question.
So, who gave theory of evolution? The options are Louis Pasteur, Charles Darwin, Robert Hooke, or Robert Brown. The theory of evolution was given by Charles Darwin. The right answer is Charles Darwin. So next question is, in which book Charles Darwin published his theory of evolution? The options are Two Sticks, Origin of Species by Natural Selection, Road Not Taken, or Evolution Theory. The answer is Origin of Species by Natural Selection is the book in which Charles Darwin published his theory of evolution. Now the next question is rapid increase in the number of species with a common ancestor. What it is called? So the options are evolution, genetic drift, adaptive radiation, or allele. So the answer is adaptive radiation. So why adaptive radiation? Adaptive radiation is you can say it is defined as when there is an rapid or very fast increase in the diversification of the species which are having the common ancestor, which happens due to the adaptation to the ecological environment, and there is a change in morphology and physiology. That is called adaptive radiation. So the next question is organism in which genetic material has been changed through biotechnology in a way that does not occur naturally by multiplication and or natural recombination. That means we have to find an organism, we have to tell what kind of organism is called in which genetic material is changed through biotechnology. There was no natural process occurred, only artificial process occurred. So we have to tell what is that organism called. So the options are genetically modified organism, or we can say GMO, clones, artificial organism, or all of the above. The answer is genetically modified organism, GMO. These are the organisms in which genetic material has been changed through biotechnology, and there is no natural changes occurred. They are changes that occurs that is artificially done in the labs, and genetic material is changed, that is DNA, by inserting a DNA or gene from other organism, and these are called genetically modified organism. So next question is technique used to make exact genetic copies. We have to make the exact means same, exact, identical genetic copies of living things. Which technique it is? The options are ELISA, cloning, biotechnology, or gene therapy. The answer is cloning. Cloning is the technique which is used to make the exact genetic copies of any living thing or living being by making clones of that organism. Next question in who is the father of biotechnology? The options are Robert Hooke, Charles Darwin, Carol Ericke, Robert Brown. The answer is Carol Ericke. So he was the one who coined the term biotechnology in 1919. Okay, so in 1919 he coined this term, and that's why he is known as the father of biotechnology. Okay, next question. So the next question is, which bacterium is used to create Bt cotton? Right. So these are the microorganisms. The option first is Bacillus cereus, Bacillus subtilis, Pseudomonas butida, or Bacillus thuringiensis. The answer is Bacillus thuringiensis. So from this bacteria, we take the gene and insert it into the plant cell, and then when the plant grows, it becomes resistant to the pests, because Bacillus thuringiensis produces, it's a bacteria, it produces a toxin which is harmful to the insects, but it is not harmful to the humans. So we can consume these genetically modified organisms, or we can say genetically modified plants, like Bt cotton, Bt brinjal, and Bt corn. These are many more we have created. We have created genetically modified organism plants that we can consume, and it is very helpful for the agriculture purpose. So the question is, which nematode infects the roots of tobacco plants, which reduces the production of tobacco? So these are the options: Melodyne incognita, Paratrichodrus alias, Cenohabditis elegans, Heterodera sacchatis. The answer is Melodyne incognita. So this is the nematode which infects the roots. So you have to remember this infects the roots of the plant. Which plant? Tobacco plant. And it reduces the production of tobacco because of this. So next is the technique that helps in early detection of diseases, pathogens, by the amplification. This is the important word, amplification of nucleic acid. So from this word you can answer which of the following technique is this. The options are polymerase chain reaction, PCR, gene therapy, vaccination, ELISA, or we can say enzyme linked immunosorbent assay. The answer is polymerase chain reaction. Polymerase chain reaction helps in the amplification of the nucleic acid, and through which we can detect the disease or pathogen, or we can say any genetic defect in the gene. So this is the right answer. And the last question is a process of cellular defense. So we have to answer the cellular defense process, in which the cells silence the mRNA due to complementary double-stranded RNA molecule. So in this cellular defense, mRNA is silenced using double-stranded RNA molecule that binds to, and it prevents the mRNA translation. So mRNA will not become the protein. So we have, and due to that, cell defend itself. Okay, so the answer, the options are replication, RNA interference, metabolism, or transcription. So The answer is RNA interference. Why? So what happens here is, whenever there is a DNA, we can say it's a DNA, a double-stranded DNA, and transcription occurs. So during transcription, mRNA is formed. Okay, so this is DNA. This is DNA, right? And this is mRNA. Right? So mRNA, so when DNA is transcribed into mRNA, this is called transcription. Sorry for the handwriting, right? So this is called transcription. And when mRNA is converted to protein, so protein is like this. This is something we call a protein. Okay, so this is what protein, and protein has some certain function. So whenever the cell needs to defend itself and needs to stop making this protein, which can be harmful for the protein, for the cell. So there is double-stranded RNA molecule which binds to this mRNA, and it stops this translation process. I can show you like this. It stops this RNA process, it silences the mRNA, and this whole process is called RNA interference. So these are our 10 questions. Please subscribe to our channel, like this video if you understood something or you understood our concepts or questions.